Here we go. Let's do some protein synthesis. Okay, so first things first. We've talked about genes before. We've talked about why DNA is awesome, but we haven't really talked about why DNA is awesome. We know that it codes for traits, but we haven't really explained why, and here's the deal. It's really, really, really simple. A gene is just a section of DNA that codes for a protein. That's it. So a gene is just a part of DNA. And what makes one gene different from another gene is the order of the A's, G's, C's, and T's. So just those bases, the adenine, guanine, cytosine, thymine, what order you put them in, that's how you get different things. For example, if you take the letter D, the letter O, and the letter G, and you put them together in a particular order, you get a dog. Awesome. If you take those same three letters and you put them together in a different order, you get G-O-D, God, a completely different thing. Unless you ask my mom, in which case they're the same thing. If you take O-G-D and put them in that order, it doesn't really mean anything. So if you take those letters and you just move them around in different orders, you get different things. That's how DNA works. Literally, the reason you are different from a banana is because of A's, G's, C's, and T's, and the order that they are arranged. The number of them you have, the order of them you have. It comes down to those four things. So if you figure our alphabet for our language has 26 letters, think of how incredible it is that with only 4 letters, our DNA does all of the things that it does, codes for all those different things. You can have thousands of genes on one chromosome. So gene, 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 chromosome, okay? Humans have about 20,000 genes, 23,000 genes, they're, mapped it out, figured it out. You've got about 23 chromosomes, so you have exactly 23 chromosomes, chromosome pairs, 46 chromosomes total, but 23 pairs, because these have code for the same things, just in different ways. So that's about 1,000 genes on a chromosome. So remember when I said gene, 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 do that 1,000 times, and that's what a chromosome is. All right, so the point of a gene is that it codes for a protein. We know already that every protein does a different job and that proteins are really, really super specific because of their shape. And so the thing you need to know is that one gene codes for one protein. That's it. Any given segment of DNA can create one protein. That's it. Okay? This gene can only code for this protein, no more, no less. Okay? This gene codes for this protein. Okay? If this protein needs to get made, this gene is the one that's going to make it. If you destroy this gene, guess what? This protein doesn't get made, okay? So one gene, one protein, incredibly specific. So it takes 2 steps to get from the gene to creating the protein. So we've talked about how these are your DNA is your blueprints, okay? That's exactly what it's going to be. The whole process altogether is called protein synthesis, but protein synthesis breaks into 2 parts. There's transcription and there's translation, and I'm going to walk you through the particulars of each one. Okay, but before we do that, we have to talk about RNA. There are 3 types of RNA. There's messenger RNA, which conveniently is abbreviated with a little m, and then of course capitalized RNA. There's transfer RNA, which is a little t and then capitalized RNA, and there's ribosomal RNA, which is a little r and capitalized RNA. You got to watch that in your handwriting. Make sure you're doing that appropriately. RNA is really similar to DNA, but it's not exactly the same. There are a handful of differences. So first of all, ribose, not deoxyribose. So RNA is ribonucleic acid, not deoxyribonucleic acid. So it's just a different sugar. So RNA has a different sugar from DNA. RNA has ribose, whereas DNA has deoxyribose. RNA is only single-stranded, whereas DNA is double-stranded. And RNA has uracil. So there's no thymine in RNA. DNA has thymine, RNA has uracil. Okay, it's a little confusing, but it gets in there. Why is it like that? It just is. That's how we evolved. We evolved to make it that way. It's not as though biology teachers at some point went, ha 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 ha, let's change the name of it and put a new molecule in. That's just the way we evolved. It's just the way it is. Okay, so transcription. Trans means across and scribe means write. So transcribe just means to write differently. So what we're doing here is we're using DNA as a template to make a strand of mRNA. Here's the reason why. We've got our DNA in the nucleus of our cell, and it's really, really important to us. If we screw up our DNA, we screw up everything our cell does. So we need to protect our DNA. That's why we keep it locked away in the nucleus. Have you ever seen the Mona Lisa? Do you know what the Mona Lisa looks like? Okay, probably. Have you ever stood in front of the Mona Lisa, actually physically at the Louvre in Paris? Maybe not. Maybe you have. You're super lucky if you have. But maybe you haven't. But you've seen it. And the reason you've seen it is because you've seen copies of it. And we've decided that the Mona Lisa is important enough for us to make that visible to, or available to the masses, to everybody to be able to see. But what if we couldn't make copies? Would it be really practical to take the actual Mona Lisa and take it from school to school to school to school so that school children across America could see the Mona Lisa? No, the Mona Lisa would end up with a mustache, a beard, and devil horns within 30 seconds. Okay, so we don't do that. We keep the really important thing, the original, protected, and then we make copies to send out into the world, because if something happens to those copies, it's no big deal. Plus, more copies can get more places at the same time. So it's just more useful that way. So that's the point of transcription, is that we want to make this mRNA copy of our gene so that we don't have to send the actual DNA out into the cytoplasm where, you know, all the riffraff and bad neighborhood is. Okay. So transcription is going to happen in the nucleus. That should make sense to you because that's where your DNA is. DNA is in the nucleus. That's where transcription is going to take place. Like I said, it makes disposable copies of mRNA to be made of our valuable originals. Transcription is really similar to replication. What we're going to have happen here is the DNA is going to unwind so that only the gene we want is exposed. So basically a signal has come into the nucleus and said, hey, we need more of this protein. Okay, so maybe we need more hemoglobin. And so the DNA is going to unwind at the hemoglobin location. The enzyme that attaches to the DNA, look at that fancy underline, I don't know why that's there. The enzyme that attaches to the DNA and reads it and puts the mRNA together is called a really logical name, and I bet you can figure it out if you think about an enzyme you knew of before. What do you think the name of that enzyme would be if it's going to make a big molecule of RNA? RNA polymerase, right? Just like there was DNA polymerase, now we have RNA polymerase. Okay, so only one side of the DNA is going to get transcribed here. There's going to be a coding side and a non-coding side. You don't need to know how to figure out which side is which, okay? Just trust me, only one side is going to get transcribed. The base pairing rules are a little bit different here. Cytosine is still going to bond to guanine, but now adenine is going to bond to uracil. Remember, we said that in RNA, there's uracil, not thymine. So adenine is going to bond to uracil. When the gene gets transcribed, the RNA is going to slide out, the DNA is going to zip back up, and the DNA is not going to have been changed at all. So it's not like replication where the DNA gets changed and you've got one old, one new. You've got the old stuff, it splits apart, RNA gets made, RNA leaves, DNA zips back up again, and we're all good. Okay, so here's our DNA. It's my Gattaca gene. It's my favorite gene to use as an example. So here's my DNA. I've got my Gattaca gene. The DNA is going to split up, and now the RNA is going to be made. Go ahead and transcribe what you think the RNA strand is going to be. I will tell you that this is the coding side. So you want to code off of that side. That's the coding side of the DNA. What do you think the mRNA strand is going to look like? I'll wait. All right, let's check and see if you were right. Okay, G bonds to C, A bonds to U now because we're talking about mRNA. T still bonds to A, T bonds to A bonds to U, C bonds to G, and A bonds to U again. And now my mRNA is going to leave and my DNA zips back up again. Okay, that's it. That's transcription. After transcription, the mRNA is going to leave the nucleus and find a ribosome. But wait. Something else happens first. The mRNA is going to get edited, and there are going to be pieces that stay inside the nucleus and other pieces that get to leave the nucleus, okay? So the mRNA is this big, long piece, and then editing needs to happen. Just like when you write an English paper and you have too much extra crap in it, and so your teacher makes you cut a bunch of stuff out, it's the same thing with mRNA. So the mRNA got created, and then the pieces that are unnecessary are going to get taken out. Weirdly, those things are called introns, okay? So introns get taken out of the mRNA, and I'll tell you why they're called introns in a second. The parts that stay in the mRNA are called exons, okay? They're introns and exons. Exons, here's why. Introns stay in the nucleus. Exons exit out of the nucleus. So introns get cropped out of the mRNA and stay in the nucleus. Exons get to leave the nucleus. They're ready for prime time, okay? So our exons are bonded back together again, and they're going to leave and go find the ribosome, okay? Why the ribosome? Why are they going to a ribosome? Good. Hopefully you remember ribosomes are where proteins are made. We learned earlier on that that's the function of a ribosome. So once the mRNA meets the ribosome, the next step is going to begin, and that's called translation. Translation is going to happen in the cytoplasm because that's where the ribosomes are. It's going to take the message from the mRNA and translate it into the actual protein. So I showed you this image before. This is a ribosome, but now we understand a little bit more about it. So all of this greenness is ribosome. And now we can see that we have the mRNA that's here because you can see here's our sugar phosphate backbone. And then here are our individual bases, our A's, G's, C's, and T's sticking out the sides. Okay, so then this, what do you think this strand is? Good, that's a protein. So then what's each one of these little things? Amino acids, because we know amino acids are the building blocks of proteins. So this is the protein chain that's getting made. The ribosome is going to read 3 nucleotides at a time, and those 3 nucleotides in a row are called a codon. So if we're looking at that mRNA molecule that we made a minute ago, GAU, UACA, what's the first codon going to be? Good, GAU. tRNA, the transfer RNA, is off floating around in the cytoplasm, and it has on it an anticodon that's going to correspond to the codon so that the tRNA can come meet up with the mRNA and they can bond to each other, okay? So then there are a bunch of different tRNA molecules out there in the world. In fact, there's at least one for every codon that can exist. Okay, so the ribosome is going to read that GAU codon, and it's going to look for a tRNA molecule that can bind with it. So what is the anticodon going to be that can bind with GAU? Hopefully you said CUA, because the G needs to bind to a C, the A needs to bond to a U, and the U needs to bond to an A. So that's the anticodon. So the CUA tRNA is carrying with it an amino acid. And this is good news because remember we said we're trying to build a protein, and proteins are built with amino acids, and so we're finally getting to amino acids, so we're going to have a protein soon. Yay! So the tRNA is carrying an amino acid, and this specific tRNA molecule is carrying an amino acid called aspartic acid. Okay? How do I know that? Magic! Because I'm the smartest. No, that's not it at all. I have a cheat sheet, and that cheat sheet is this thing called the genetic code. So I like this one because it's a wheel, but you'll find other ones online that are boxes, but I think this is the easiest kind to follow. Okay, so what we're going to do is we're going to look at our codon. Now, this is important. We look at the codon. The codon, the codon. So we're looking at the mRNA. We are not looking at the tRNA. We are looking at the mRNA when we use this. So we look at the codon. Okay, so the first letter of our codon was G, so we start in the center of the wheel at G. Okay, the second letter in our codon was A, so we move up from the G to the A. Then the third letter was a U, so we move from the A out to the U. Okay, this now tells us that the amino acid we're looking at is aspartic acid. Okay, so basically you start at the center and then you work your way out in whichever direction you need to work your way. All right, so while holding on, the ribosome is going to move down to the next codon. So what's the next codon in our example? UAC. What's the corresponding tRNA anticodon to that? AUG. What amino acid is this tRNA molecule going to carry? Tyrosine. Okay. Tyrosine. If you couldn't figure out where I got tyrosine from, go back to the slide where I showed you how to use the genetic code and figure it out, because this is important. You have to be able to do this. Okay, the ribosome then is going to hook together the 2 amino acids. How's it going to hook those 2 amino acids together? What's it going to use? Good, a peptide bond, because we know that amino acids are connected to each other with a peptide bond. Okay, so now we have our aspartic acid that's bonded to tyrosine. The ribosome is going to move down the mRNA strand in 3 nucleotide chunks until it gets to a codon that says stop. Shockingly enough, this is called a stop codon. Now here's something important. The stop codon does not code for an amino acid. It just literally stops translation and makes the ribosome let go of the mRNA. It does not put anything onto the end of the protein. Okay? Nothing gets stuck on there. There's no caboose. There's no stop sign at the end. There's no cap at the end. The stop codon literally just means stop. So when you're telling me the amino acid sequence of a protein, do not write stop at the end of it because there's no stop amino acid. It's not proline, cytosine, aspartic, or proline, aspartic acid, tryptophan, stop. Okay? It's just those 3 and then that's it. It's done. Okay? After translation, the mRNA might go find another ribosome. The ribosome might go find some other piece of mRNA. Everything's totally recyclable in this process. Now, just to complicate matters, translation doesn't start at the first nucleotide. And translation doesn't even necessarily start at the first codon. Translation is going to start at the first AUG codon. So the ribosome is going to read along a gene, read along a piece of mRNA, all the way until it reaches the AUG codon. Okay? So until there's an AUG codon, everything else gets ignored. Okay? Everything gets ignored before that point. Okay? What does AUG code for? Good, methionine. So every single protein that we ever have starts with methionine. Methionine is the starting amino acid. AUG codes for methionine. Nothing will start until AUG. So everything's going to start with methionine. All right. So let's give it a try. You're going to get lots of practice here. Okay. So our ribosome is going to attach here. We're looking at a GCU codon. Is anything going to happen? No, nothing's going to happen. Here's the CUC codon. Anything going to happen? Nope. UCA. Nope, still not there. CAU. Still not there. AUG. Yay! I'm not going to make you look up what AUG is because I just had you look up what AUG is. Okay, so what's going to happen now is tRNA is going to come in and tRNA is going to bring methionine with it. So that amino acid comes in and the tRNA is going to bond to the mRNA and the ribosome is making that happen. So there's one kind of RNA we haven't talked about yet. That's the ribosomal RNA. Ribosomal RNA is in the ribosome. So the ribosomal RNA is kind of like a matchmaker. It's going to connect the mRNA and the tRNA and make them be friends. Okay? Now the ribosome is going to move down. Now here's one thing I didn't tell you about ribosomes. Ribosomes have 2 sites. There's a P site and an A site. Okay? The A site is this one. It's sort of the working site that's going to go looking for the tRNAs. The P site is a holding site. This one's just holding on. It's like, hey, I'll hold on to this guy for you and not let it go away while you go find what belongs here. Okay? So we have a CGA codon. What is the anticodon going to be for that? So in other words, what's going to be on the tRNA molecule in order to bond to this? Okay, good. And then what amino acid will that tRNA be bringing to us? Be careful. Okay, hopefully you said arginine. Now notice that when arginine came in, a peptide bond was formed here. So we have a peptide bond between our 2 amino acids. Now the ribosome is going to move down a spot. So now the arginine tRNA is in the P site and the A site is looking for something else. And then there's this other site called the exit site, the E site. And the E site is going to allow that tRNA to leave so that it can go off and find another amino acid that maybe we'll need later. Okay? So now we have UUC. What's the anticodon that's going to be on the tRNA molecule that comes over for UUC? Okay, good. And what is the amino acid that's going to come over then with that tRNA? Hopefully you said phenylalanine. Okay, so phenylalanine attaches and now we have another peptide bond. Okay, peptide bond forms The ribosome moves down. We now have this guy in the P site, the A site waiting for a new thing, and then in the E site, that tRNA is going to leave, okay? GAU, you know the routine. What is the tRNA anticodon? Excellent. And what's the amino acid? Good. Aspartic acid. We saw that one before. Okay, now the ribosome is going to move down. Okay, the tRNA is going to leave. And now we have UGA. What is UGA code for? Good. UGA is a stop codon. So what's going to happen? Is the caboose going to come in? No. Is the stop amino acid going to come in? No. Nothing's going to come in. Things are just going to let go. And so now we have mRNA separate from our protein that we've just created. So we're finished now. Our ribosome can go off and do something else. Our mRNA can go do something else. Our protein can go be a protein. And our tRNAs can go off and find more amino acids. Okay? So to sum things up, transcription happens in the nucleus. It changes the message from DNA form to RNA form. Be really careful. It doesn't change DNA into RNA. Okay? It's not like we take DNA and go, blah, 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 look, RNA. We take the message, and we take the message out of the DNA form, and we make an RNA copy of that message. And then translation is going to happen at the ribosome, which is in the cytoplasm, and it's going to use the mRNA message to make a protein. Okay? So transcription makes the mRNA from DNA to carry the message out to the cytoplasm, where translation makes the protein. There you go. Simple, simple, simple.